Hello, everyone. Welcome back to Meds.Concepts. Today we are going to discuss only basics of DNA replication. We will discuss the prokaryotic and eukaryotic DNA replication in a separate video. DNA replication is the process by which a cell produces an identical copy of its DNA before cell division. Without accurate replication, genetic information cannot be transmitted properly from one generation to the next. This topic is extremely important for CSIR-NET because many conceptual and mechanism-based questions are asked from replication, proofreading, DNA polymerase and replication enzymes. In this lecture, we will study models of DNA replication, Meselson and Stahl experiment, replicons origins, replication forks and replicons, requirements, DNA polymerase, proofreading, primase, fidelity mechanisms, and types of prokaryotic DNA polymerase. So let's start. DNA replication occurs during the S phase of the cell cycle. The primary objective of replication is to ensure that each daughter cell receives an identical set of genetic information. Replication is semi-conservative in nature. This means each newly formed DNA molecule contains one parental strand and one newly synthesized strand. DNA replication requires several essential components: template DNA, primer, DNA polymerase, DNTPS, Mg2+ ions. An extremely important point is that DNA synthesis always occurs in the 5'3' direction. DNA polymerase adds nucleotides only to the free 3' group of the growing strand. Replication is both template-dependent and primer-dependent. Without primer, DNA polymerase cannot begin synthesis. Models of DNA replication. Before the mechanism of replication was experimentally proven, scientists proposed 3 different models. The first model was the conservative model. According to this model, the parental DNA molecule remains completely intact, and an entirely new daughter duplex is synthesized. The second model was the semi-conservative model. In this model, each daughter DNA molecule contains one parental strand and one newly synthesized strand. The third model was the dispersive model. According to this model, parental and newly synthesized DNA are interspersed in both strands. Among these models, semi-conservative replication was later proven experimentally by Meselson and Stahl. The dispersive model remained only theoretical and is not observed in normal cellular replication. However, some specialized recombination-dependent mechanisms may resemble conservative replication in certain organisms. Meselson and Stahl experiment. This experiment is considered one of the most elegant experiments in biology. Meselson and Stahl used E. coli grown in a medium containing heavy nitrogen isotope, nitrogen-15. As a result, the bacterial DNA became labeled with heavy nitrogen. The bacteria were then shifted to medium containing normal nitrogen-14. DNA samples were collected after each generation and separated using cesium chloride density gradient centrifugation. In the first generation, only one intermediate density hybrid band appeared. This ruled out the conservative model because conservative replication should have produced one heavy band and one light band. In the second generation, one hybrid band and one light band appeared. These observations perfectly matched the semi-conservative model. Thus, the experiment conclusively proved that DNA replication is semi-conservative. Replicons and origins of replication. A replicon is a segment of DNA replicated from a single origin of replication. In prokaryotes, Usually one chromosome contains a single origin. Therefore bacterial chromosomes are generally monorepliconic. In eukaryotes, chromosomes are very large. Therefore multiple origins of replication are required. As a result, eukaryotic chromosomes are multirepliconic. Each replicon in eukaryotes may range approximately from 40 to 100 kilobases. The origin of replication is the specific DNA sequence where replication begins. Initiator proteins recognize these origins and recruit replication machinery. Multiple origins allow rapid replication of large eukaryotic genomes. Replication fork and replication bubble. When DNA replication begins at an origin, the double helix unwinds. This creates a Y-shaped structure known as the replication fork. In bidirectional replication, two replication forks move in opposite directions from the origin. The region between the two forks is called the replication bubble. Most organisms show bidirectional replication because it increases replication efficiency. Unidirectional replication is comparatively rare. At the replication fork, helicase unwinds DNA. Single-strand binding proteins stabilize strands. Topoisomerases remove supercoiling. And DNA polymerase synthesize new DNA. DNA replication requires several essential molecular components. First is template DNA. The parental DNA strand provides sequence information for synthesis of the complementary strand. Second are DNTPs, DATP, DGTP, DCTP, and DTTP. These act as substrates and energy sources. Third is the primer. DNA polymerase requires a free 3' group to initiate synthesis. Fourth is DNA polymerase itself, which catalyzes phosphodiester bond formation. Fifth are Mg2+ ions, which are essential cofactors for catalysis. Without Mg2+, polymerase activity cannot occur efficiently. A very important concept is that DNA polymerase cannot initiate synthesis de novo. DNA polymerase. DNA polymerase are enzymes responsible for DNA synthesis. They possess multiple enzymatic activities. The most important is 5' to 3' polymerase activity, which synthesize DNA. Many polymerases also possess 3' to 5' exonuclease activity, which performs proofreading. Some polymerases such as DNA polymerase I additionally possess 5' to 3' exonuclease activity, used for primer removal. DNA polymerase are template-dependent, highly accurate, and possess a single catalytic active site. Other enzymes related to nucleic acid synthesis include reverse transcriptase, primase, RNA polymerase, and terminal transferase. DNA synthesis always proceeds in the 5' to 3' direction. The mechanism of DNA synthesis involves nucleophilic attack. The free 3' group of the growing DNA strand attacks the alpha phosphate of the incoming DNTP. This forms a phosphodiester bond between nucleotides. During this reaction, pyrophosphate is released. Hydrolysis of pyrophosphate provides thermodynamic drive and makes the reaction essentially irreversible. DNA elongation therefore occurs only at the 3' end. Because DNA strands are antiparallel, one template strand runs 3' to 5' while the complementary strand runs 5' to 3'. This antiparallel arrangement is essential for replication. Fidelity of DNA replication. DNA replication is highly accurate. The error rate is approximately 1 mistake per 10 to the power of 9 to 10 to the power of 10 nucleotides added. This remarkable fidelity results from several mechanisms. First, correct hydrogen bonding between complementary bases improves specificity. Second, the active site geometry of DNA polymerase allows only correctly paired bases to fit efficiently. Third, proofreading activity removes incorrectly inserted nucleotides. Another important feature is steric discrimination. DNA polymerase strongly discriminate against RNTPS because ribonucleotides poses a 2' group. As a result, RNTPS are incorporated approximately 1,000 times slower than DNTPS. Role of metal ions and tautomerism. DNA polymerase require divalent metal ions, especially Mg2+. The two-metal ion mechanism involves metal ion A, which activates the primer 3' group, and metal ion B, which stabilize negative charges on phosphate groups. Another important concept is tautomerism. Nitrogenous bases normally exist in keto and amino forms that allow standard base pairing. A with T and G with C. Rare tautomeric shifts may alter hydrogen bonding patterns. For example, enol-guanine may pair with thymine instead of cytosine. Such abnormal pairing can introduce mutations during replication. Therefore, tautomerism contributes to spontaneous mutation formation. Proofreading activity. Proofreading is an extremely important fidelity mechanism. Many DNA polymerase poses 3' to 5' exonuclease activity. When an incorrect nucleotide is inserted, polymerase progression slows, the newly synthesized strand shifts into the exonuclease site, and the incorrect nucleotide is removed. After removal, the strand returns to the polymerase site and correct synthesis resumes. Proofreading greatly increases replication fidelity by approximately 102,000-fold. An important point is that polymerization and proofreading occur at separate catalytic sites. Primer and primase. DNA polymerase cannot start synthesis independently because it requires a pre-existing 3' group. This requirement is fulfilled by primers. Primers are synthesized by primase, specialized RNA polymerase. Primase produces short RNA primers complementary to the DNA template. On the leading strand, only one primer is required because synthesis is continuous. On the lagging strand, synthesis occurs discontinuously, forming Okazaki fragments, therefore multiple primers are needed. These RNA primers are later removed and replaced with DNA. Prokaryotic DNA polymerase. In E. coli, 5 major DNA polymerases are known. DNA polymerase 1 removes RNA primers, fills gaps, and participates in repair. It possesses 5' to 3' polymerase, 3' to 5' proofreading, and 5' 3' exonuclease activities. DNA polymerase 2 mainly functions in DNA repair. DNA polymerase 3 is the primary replicative enzyme. It possesses high processivity, high speed, and proofreading ability. DNA polymerase 4 and 5 participate in SOS repair and translesion synthesis. These polymerases are more error-prone but help cells survive DNA damage. Finally, let us revise the most important points. DNA replication is semi-conservative. Replication always proceeds in the 5' to 3' direction. DNA polymerase requires template, primer, DNTPS, and Mg2+ ions. Replication bubbles contain two replication forks. Proofreading uses 3' to 5' exonuclease activity. DNA polymerase 1 removes primers. DNA polymerase 3 is the main bacterial replicative polymerase. Tautomeric shifts may cause spontaneous mutations. Primase synthesize RNA primers. These concepts are highly important for CSIR-NET and other competitive examinations. Thank you. If you found this video helpful, please like, subscribe and share this to your friend.